developed first. It is bilaterally symmetrical. It has 3 tissue layers, the endoderm, ectoderm, and mesoderm. It has no respiratory system, which is why it can hold its breath for very long amounts of time. Its body cavity is a hemocoel, which is similarly to a circulatory system. And it has 4 pairs of legs with 8 claws.
Creatures only vary in length from 0.1 to 1.2 millimeters. Most species are found in the freshwater, but they can also be found all over the world, from tropical rainforests to the Appalachians, the ocean, and Antarctica. They can be even found in the moss in your own backyard or algae in the gutters.
Tardigrades come from the Latin word tardis for slow, and gradu meaning a step, which means they are slow walkers, which they are. They walk using side-to-side, lumbering, and Ursan movements. Oh my gosh, a rare sighting of a tardigrade!
Tardigrades are sartorial feeders. They have 2 sharp stylets within the mouth, which they use to pierce the cells of the food, and the fluids are then sucked out of the cell using the muscular pharynx as a pump. The diet includes bacteria, algae, and decaying plant matter. Larger species are known to feed on smaller tardigrades. Yes, folks, they are cannibals.
They reproduce sexually similar to humans, with the females laying eggs and males producing sperm. However, in some species, only females have been found, so these species are assumed to be asexual.
Tardigrades have an interesting life history. They're direct, meaning they have no larvae, and their juveniles hatch from eggs. The number of cells in a tardigrade's body is fixed from birth. Although tardigrades don't migrate intentionally, the small size of their eggs in tough forms allow them to disperse by wind and bigger animals.
There are 3 classes of tardigrades. The first one is U. tardigrada, and they're primarily found in fresh water and have a smooth external cuticle. The second class is Heterotardigrada, which is distinguished by the gonoducts that open to the outside. The third class is Mesotarda. It only contains 1 species, but the location of the primary sample was destroyed in an earthquake, so the status of this class is uncertain. Imagine the toughest animal alive. A tiger? A shark? Nope, you're wrong. It's a tardigrade. They can survive many environmental extremes by converting themselves into a ton, also known as cryptobiosis. This involves them pulling in their legs and forming a cylinder-like structure, and then shutting down all their metabolism. Any other animal that goes through this process is considered dead. Only tardigrades can return to this active state.
There are 4 types of cryptobiosis they can go through. The first one is anhydrobiosis. It's when they have a loss of water in the tardigrade's environment. Then there's osmobiosis. That's when there's an increased amount of salinity in the environment. And then there's cryobiosis. That's when there's a low temperature in its environment. And finally, anoxybiosis, when there's a lack of oxygen in its environment. For this reason, scientists have tested the survival of tardigrades, and they can survive in high X-ray radiation or even the vacuum of space. We constructed 2 trees, one from our data matrix in the lab and one using 16S rRNA sequencing. These are some of the characteristics that evolved over time in these phylum. The closest relative to the tardigrada is mollusca, but in the data matrix tree, the closest relative to the tardigrada is the annelida. This is probably due to the characteristics that we chose, which were number of hearts, habitat, and eyes.
This is a tree we found in a journal article of tardigrade evolutionary history using molecular phylogenetics. Tardigrades have been thought to be a most ancient lineage, having evolved in the Cambrian period 530 million years ago. However, it has been difficult to determine exactly where tardigrades should fall on a phylogenetic tree due to the difficulty of finding fossils and their evolutionary history due to size and soft bodies. However, scientists place tardigrades in close relations to arthropods, mollusks, and nematodes.
We predict that the trees generated looks different from our observations because they use 18S rRNA to analyze the evolutionary history of the tardigrade. They also used more sister species in the study to get more accurate results. They also use a different outgroup for the trees, so it affects how the tree generates. Thanks for tuning in today's broadcast, and see you next week.